Welcome back to medforyou. Today, we will be diving into the topic of SGK1, a fascinating gene that impacts our health in numerous ways. So, let's dive in. To start off, cerebral glucocorticoid regulated kinase 1 or simply SGK1 is a gene that regulates cellular stress response, cell survival, and renal sodium excretion. It also controls processes like hormone release, inflammation, and cell proliferation. High levels of SGK1 expression are linked to conditions such as hypertension and diabetic nephropathy. SGK1 frequently mutates in diffuse large B-cell lymphoma, which led scientists to believe that SGK1 acts as a tumor suppressor. This gene also helps regulate diverse cerebral diseases such as Parkinson's disease, schizophrenia, and depression. A groundbreaking study by Gu et al. Revealed that DLBCL patients with multiple SGK1 mutations had a significantly better prognosis compared to those without. This study conclusively demonstrates that SGK1 mutations serve as a positive prognostic marker for DLBCL. Interestingly, mutations of the SGK1 gene may be an example of natural selection, where people with the mutated SGK1 proteins have an increase in the survivability rate against cancer. Moreover, SGK1 is identified as a member of the AGC subfamily of protein kinases. This subfamily includes other kinases such as PKA, PKG, and PKC, all of which share a high homology and catalytic kinase domain. SGK1 has a serine or threonine kinase activity. Moreover, it was first identified as an immediate relay gene activated through the process of transcription by serum and glucocorticoids in rat mammary tumor cells. SGKs are considered to be one of the most evolutionarily conserved groups of protein kinases within the eukaryotic kingdom. The SGK family comprises of three isoforms, namely SGK1, SGK2, and SGK3, which are highly similar to each other but are encoded by different genes located on separate chromosomes.
A study using a high-fat diet-induced mouse model was conducted to investigate the role of SGK1 in promoting Alzheimer's disease-associated tau pathology. The activation of SGK1 in the brain was found to increase tau phosphorylation and aggregation. Inhibiting SGK1 activity in HFT-fed mice reduced tau phosphorylation and improved cognitive function, suggesting that SGK1 may be a potential therapeutic target for treating AD-associated tau pathology. Additionally, SGK1 has the ability to directly increase ENaC levels by phosphorylating the channel protein and indirectly by phosphorylating medforyou, a protein involved in ENaC degradation. This regulation of ENaC activity has been associated with hypertension and insulin resistance, common features of type 2 diabetes. SGK1 mutations have been shown to impact proteins involved in insulin signaling such as AKT and FOXO1, potentially contributing to the development of insulin resistance in type 2 diabetes. Targeting SGK1 in future treatments may enhance insulin sensitivity and glycemic control in individuals with type 2 diabetes. Furthermore, SGK1 activates GSK3 beta, which phosphorylates tau protein, leading to its aggregation and the formation of neurofibrillary tangles, a hallmark of Alzheimer's disease. This suggests that SGK1 gene mutations may promote the development of Alzheimer's disease by increasing tau phosphorylation and aggregation. Now that we know the origin and how this gene mutation operates, you might be asking, what is the relevance of this information? Well, ever since we understood the mechanism of SGK1 gene mutation, scientists have developed potential treatments of cancer and discovered how this gene is apparently a key player in other neurological diseases aside from Alzheimer's. SGK1 as a gene expression regulator controls cell proliferation. In fact, SGK1 was found to be expressed highly on tumor cells. Thus, researchers today are interested in inhibiting the activity of SGK1 to prevent cancer cell growth. An example of an inhibitor of this gene, QGY5114A, was recently developed in 2019 by Liang et al. The SGK1's role in neuronal development has also been made clear. It was observed that neuronal proliferation and differentiation of human hippocampal progenitor cells was reduced by SGK1 gene activity. The high cortisol levels associated with this gene also hints its potential use in determining the risks of heart disease and depression. Indeed, the field of molecular medicine has been expanding. Thanks to the clearer vision of the mechanism of this gene mutation, we are discovering new approaches for treating cancer and therapies for mental disorders. And that's a wrap for today's gene, the SGK1. We hope that you learned something valuable today, and as always, stay curious!